So this video is a video that will help you guys with the DNA replication lab. So normally we would be in school doing this, but if we're not, this is going to have to suffice. So this diagram is showing you a replication fork. Replication fork is an opening in DNA that has been opened by DNA helicase. So this would have been closed and it's opening in the direction that way, from right to left. So we're going to start with our replication fork slightly opened and put our DNA helicase on the part of the DNA double helix that isn't yet opened. So helicase would be acting right there and opening it, and you can see the little arrow showing you the direction pointing that DNA helicase would be acting. So now the next thing we need to do is figure out which is our leading and lagging strand. So now, if this template strand is 5', the new strand is going to be built anti-parallel to the original strand. So this is going to be 3', and if the bottom strand, if the original template is 3', the new strand will be 5'. So if you were thinking that the leading strand will be on the bottom, you would be correct. So next we put in our next enzyme, and it's DNA polymerase. And we're putting our DNA polymerase where it would start, so it's moving in the 5' to 3' direction. And the first nucleotide in our double helix is in the window. And this is on the leading strand. So this, the complementary base to A would be T, and now we put down our DNA polymerase on our lagging strand, but polymerase can't work in the 3' to 5' direction. It can only work in the 5' to 3' direction. So it has to wait until the replication fork is open, and it has to jump ahead and build backwards that way. So the base that is complementary to C would be G. And now we're alternating. We go to the bottom, we slide our polymerase, and T goes with A. A is complementary to T. And then this moves this way. T is complementary to A. Then this polymerase moves this way. We have C next, so G is our complementary base. And now we move this way. C is our complementary base. We move this way. G is our complementary base. And we move it this way. C is our complementary base. We move this polymerase one more. T is complementary to A. Move this one more. T is complementary to A. Move this one more. We have G in the window, so C would be our complementary base. And then we go to here. T is complementary to A. And if we go to here, C is complementary to G, so our new base, our new nucleotide would come in so that guanine and cytosine always pair and thymine and adenine always pair. So now I'm just going to remove the enzymes for a minute and just build the sugar phosphate backbone. It would be happening all along, but I can't draw the sugar phosphate backbone while the polymerase is there, because in this model it's the paper enzyme is blocking it. Okay, so the first thing that I want to do is make sure that I'm drawing an arrow in the right direction on my Okazaki fragment. So this one was built in this direction. This is built in this direction, but I'm not done. So this arrowhead will tell you the direction we were going. So now we are going to do part 2, where we remove this and we open up the replication fork even more, and our DNA helicase would go there. By the end of our simulation, We tape down our DNA helicase right here, and I'm taping it down so I can lift it up when I need to label things later. So that's a final position of our DNA helicase. Now, on the upper, or on the bottom part, the polymerase just keeps going the same direction it was going. On this side, the DNA polymerase has to jump ahead and work backwards again. So we're going to go, A is complementary to T, A is complementary to T, G is complementary to C, T is complementary to A, T is complementary to A, C is complementary to G, A is complementary to T, G is complementary to C, G is complementary to C, C is complementary to G, G is complementary, C is complementary to G, T is complementary to A, G is complementary to C, oops, A is complementary to T, A is complementary to T over here, C is complementary to G, T is complementary to A, A and T are complementary to each other, and G and C are complementary to each other. Now, this is going to be where our simulation stops. I'm going to label some things. So this would be where our DNA polymerase would stop on the lagging strand. This is where it would stop on the leading strand. And I have taped it down so that I can lift it up to label things. Now, of course, I'm going to continue with our sugar phosphate backbone. And of course, in reality, this would have been built all along. And it's a sugar phosphate backbone because it's built of alternating sugar and phosphates. Now this one was built in this direction, so I'm going to actually put the sugar phosphate backbone in the direction that it was built, so I can be a little bit more authentic. Now notice there is a break right here. So this is our leading strand, and I'm putting an arrow here to show you that it went this way from right to left, and this one was built from left to right. And so this is our leading strand. And this up here is our lagging strand. Notice that I'm labeling the inner strands. The outer strands are the template strands. They can't be considered leading or lagging. They're already built. This is called the lagging strand because it's built more slowly, because the polymerase has to wait until the helicase opens the replication fork so it can jump ahead and build backwards. On the lagging strand, on the leading strand, the leading strand is here, the polymerase can follow right behind the helicase. So as the helicase is moving, the polymerase moves right behind it. So right here, would be where the enzyme seals the break, and that enzyme would be called ligase. We want to be more specific and call it DNA ligase. And this is Okazaki fragment 1, because it was the first one built, because this has been open longer. And this is Okazaki fragment 2. And the last thing to do is to label the 5' and 3' ends. So this one is 5', this is 3' here. This right here would be 5', and then to 3', because that's the end of Okizaki fragment, and this is 5', and this, well, we already labeled the 3' here, so we don't need to label that because I started with those labels. So that is the end of the simulation of DNA replication.